Cell biology, study of cell structure and reproduction, also function, is called cell biology. Cytology: study of structure. Historical background of cell. Robert Hooke first time discovered cell in Cork cell. Non-living: A.V Leunhook discovered live cell. He is also called father of microbiology. Robert Brown discovered nucleus in 1831. J.E Purkinje discovered protoplasm in 1839. Schledin discovered that all plants are composed of different little cell and form tissue in plant. Schwan discovered that cells had outer thin layer. Today we know plasmomembrane. The bodies of animals and plants are composed of cell and products of cell. He concluded the presence of cell wall is unique character of plant cells. Cell theory in 1840. A cell theory which was given by two scientists, Schledin, who was a botanist of German, and Schwan, who was zoologists of British. In this mentioned that all cells are composed of organelles and all living and non-living organisms are composed of cell. Rudolf Virchow first time explained cell division in 1855. New cells are formed by pre-existing cell. Omnicellular, acellular, those animals which are composed of single cell called unicellular organisms. Those animals which are composed of more than one cell, called unicellular organisms. Prokaryotes: the cell do not have membrane-bound nucleus. Other organelles like a lysosome, vacuole do not have membrane-bound structure. Cell division occurs either by fission or budding. Single chromosome is present. Bacteria, blue-green algae, cyanobacteria, mycoplasma, are prokaryotes. Eukaryotes: the cells have membrane-bound nucleus. In it membrane-bound structure is present. Cell division takes place by mitosis or meiosis. More than one chromosome are present. Eukaryotes: Protista, fungi, plants and animals. Three model come to describe the cell membrane or biomembrane. 1. Sandwich or trilaminar model given by Davison and Danelli in 1930. Total thickness of membrane is 80 n.m. Lipid 6.0 n.m and protein 1.0 nm. Each thickness of protein is 28 degrees. The overall thickness of lipid is 75 degrees. It is amphipathic membrane because it has two types of part. This structure is called FPLLP protein, lipid protein. Thickness of protein layer is 35 degrees. Hydrophobic tail attached to each other by van der Waals force. Hydrophilic head attached to protein through hydrogen and ionic bond. Thick model was rejected because it fails to explain fluidity and selectivity permeability of the membrane. Unit membrane model by Robertson in 1965. All the cellular and organelles membrane are similar in structure and function. This was rejected because it fails to explain fluidity and selectively permeability of the membrane. Fluid mosaic model by Singer and Nicholson in 1972. This is most widely used and accepted model for the plasmomembrane. Protein iceberg in the sea of phospholipid. Gulab jamun in the concentrated solution of sugar. Cell membrane: there are two types of protein present in membrane. One is found in periphery, which is extrinsic protein, and another one is found inside the membrane, which is called intrinsic protein. The main component of cell membrane is phospholipid. Cell membrane is living and semi-permeable. Many molecules move across it without any expenditure of energy is called passive transport. Few ions or molecules move across the cell membrane from high concentration to lower concentration is called osmosis. Some molecules move across the cell by the expenditure of energy is called active transport. In it, molecules move from lower concentration to higher concentration. Example equals Na plus K plus pump. The main function of cell is membrane is movements of molecules across it. The lipid provide fluidity to cell membrane. The quasi-fluid nature of lipid enable the movement over the protein layer. There are two types of movement present in cell membrane. One is transition movement and another one is flip-flop movement. Phospholipid solution of sugar is called phospholipid. It is main components of plasmamembrane. It gives frame structure to the plasmamembrane. It provide fluidity to cell membrane. Protein on the basis of ease of extraction. It is divided into two part. 1. Intrinsic protein inside the cell membrane. 2. Extrinsic/peripheral protein outside the cell membranes. Enzymatic activity. It's found in extrinsic protein. Cholesterol. It provides stability to the cell membrane. It is more rigid than phospholipid. In prokaryotes, cholesterol is absent in place of cholesterol, haploids is present. If carbohydrates are attached with lipid called glycolipids. If carbohydrates is attached with protein is called glycoprotein. Endocytosis is taking in material by living cell. It are of two types: 1. Pinocytes: ingestion of liquid by the cell membrane. 2. Phagocytosis: ingestion of solid material by the cell membrane. Exocytosis: cell vomiting, ingestion of waste material.
You will get notes of the related topic which has been described in the video. You will watch next video to get detail about the organelles. Please subscribe our channel for more video. Please subscribe.